CRISPR-Cas9. In clinical medicine. Challenges. Approved therapies. Future directions. Part 1. Challenges in clinical applications. What is CRISPR-Cas9? CRISPR-Cas9 is a gene editing technology. It allows scientists to cut DNA at specific locations. This makes it possible to remove, disable, or modify genes. In the lab, CRISPR is fast, cheap, and efficient. In patients, it becomes far more complex. Why clinical use is difficult? Human medicine is not a controlled experiment. Every patient is genetically unique. Every error can be permanent. Clinical gene editing must be extremely precise, highly predictable, long-term safe. This sets a very high bar. Off-target effects. CRISPR uses a guide RNA to find a DNA sequence, but the human genome contains billions of bases. Some DNA regions look very similar. Cas9 may cut the wrong location. This is called an off-target effect. Why off-target effects are dangerous? Unintended DNA cuts can damage essential genes, activate cancer-related genes, cause chromosomal rearrangements. Even rare errors are unacceptable in medicine, especially in long-lived cells like stem cells. DNA repair is unpredictable. After DNA is cut, the cell repairs it. There are two main repair pathways: fast but error-prone repair, precise but inefficient repair. Most adult cells do not divide. This limits precise gene correction. Resulting problem. Scientists cannot fully control how DNA is repaired, which mutation is created, how consistent results will be. This unpredictability is a major barrier. Delivery challenges. CRISPR is not a single molecule. It includes Cas9 protein or RNA, guide RNA, sometimes repair templates. All components must reach the correct cells. Delivery methods. Current delivery approaches include viral vectors, lipid nanoparticles, ex vivo cell editing. Each method has limitations: immune reactions, limited targeting, size restrictions. Immune system barriers. Cas9 comes from bacteria. Many people already have immunity to it. The immune system may attack edited cells, cause inflammation, reduce treatment effectiveness. This is especially problematic for repeated treatments. Ethical and regulatory challenges. Gene editing raises ethical questions. Should edits be heritable? Who gets access? Where is the line between therapy and enhancement? Regulators require extensive safety data, long-term monitoring, strict oversight. Summary of challenges. CRISPR clinical use faces precision limits, delivery barriers, immune responses, ethical concerns. Yet progress continues. Part 2. Approved CRISPR therapies, from research to medicine. For years, CRISPR existed only in labs. Recent advances changed this. CRISPR has now entered real clinical practice. The first therapies are approved for human use. Why blood disorders came first. Blood diseases are ideal targets because blood stem cells are accessible. Cells can be edited outside the body. Corrected cells can repopulate blood. This approach is called ex vivo gene editing. Diseases treated so far. Approved CRISPR therapies focus on sickle cell disease, beta thalassemia. Both are caused by single gene mutations. How ex vivo CRISPR therapy works. Step 1. Blood stem cells are collected from the patient. Step 2. CRISPR edits genes that control hemoglobin production. Step 3. Edited cells are returned to the patient. Why this strategy works. Instead of fixing the faulty gene directly, CRISPR reactivates fetal hemoglobin. Fetal hemoglobin carries oxygen efficiently, prevents disease symptoms. This bypasses the original mutation. Clinical outcomes. Patients show fewer pain crises, reduced hospitalizations, elimination of blood transfusions. Benefits can last for years. Why these therapies succeeded. Key reasons. Clear disease mechanisms, accessible target cells, measurable outcomes, controlled ex vivo editing. This proved CRISPR can be safe and effective. Limitations of current therapies. Despite success, challenges remain. Very high cost, complex hospital procedures, chemotherapy conditioning, limited global access. These treatments are not yet widely scalable. What this means. CRISPR has crossed a critical milestone. It is now an approved medical technology. But this is only the beginning. Part 3. Future directions in medical research. Beyond traditional CRISPR. Standard CRISPR cuts both DNA strands. This increases risk. New tools aim to edit genes without cutting DNA. Base editing. Base editing changes a single DNA letter. Does not create double-strand breaks. Reduces unintended damage. This improves safety and precision. Prime editing. Prime editing works like search and replace for DNA. It can correct mutations, insert new sequences, remove unwanted DNA, all with higher accuracy. In vivo gene editing. Future therapies aim to edit genes inside the body. Target organs include liver, eye muscle, brain. This would eliminate complex cell extraction procedures. Advances making this possible. Key innovations include lipid nanoparticles, tissue-specific targeting, temporary Cas9 activity. These reduce immune and safety risks. Expanding disease targets. Future CRISPR applications may include cancer treatment, neurodegenerative diseases, heart disease, viral infections, autoimmune disorders. CRISPR can modify how cells behave. Cancer and immunotherapy. CRISPR can engineer immune cells, improve tumor recognition, reduce immune exhaustion. This may create more powerful cancer therapies. Precision and personalized medicine. Every patient's mutation is unique. CRISPR allows customized genetic treatment. Medicine is shifting from one drug for all to one therapy per patient. Ethical responsibility going forward. Future success depends on global regulations, ethical boundaries, fair access, public trust. Scientific power must be used responsibly. Final message. CRISPR-Cas9 is reshaping medicine. It faces major challenges. It has already saved lives, and its future potential is enormous. The journey from DNA editing to curing disease has only just begun. Part 4. Safety, Ethics, and Societal Impact. What makes CRISPR different? CRISPR does not treat symptoms. It changes the biological code itself. This means effects can be permanent. Changes may persist for life. Mistakes cannot be undone. This makes safety and ethics central issues. Somatic versus germline editing. There are two types of human gene editing. Somatic editing affects only the treated person. Changes are not inherited. Germline editing alters eggs, sperm, or embryos. Changes are passed to future generations. Most current medical research uses somatic editing. Why germline editing is restricted? Germline editing affects people who do not yet exist. Future generations cannot give consent, cannot opt out, must live with the consequences. This raises fundamental ethical concerns. Scientific risks of germline editing. Our understanding of genetics is incomplete. Single genes often affect multiple traits, multiple biological systems. Changing one gene may have unintended effects, delayed consequences, effects that appear decades later. Irreversibility of germline changes. Once a genetic change enters the population, it cannot be recalled. It cannot be corrected easily. It may spread through generations. This makes germline editing uniquely risky. Current global consensus. Most countries prohibit germline editing, allow somatic editing under strict rules. International scientific organizations call for extreme caution, global cooperation, strong ethical oversight, long-term. Safety in somatic editing. Even non-heritable editing raises safety concerns. Gene-edited patients may face delayed side effects, increased cancer risk, unexpected immune reactions. Some risks may not appear for years. Need for lifelong monitoring. CRISPR-treated patients require long-term follow-up, continuous health monitoring, genetic surveillance. This is different from traditional drug therapies. Cancer risk and genomic stability. DNA breaks can cause chromosomal rearrangements, activate cancer-related pathways. Even rare events matter when millions of cells are edited. Risk versus benefit in severe disease. Ethical decisions depend on context. For life-threatening diseases, higher risk may be acceptable. For mild conditions, risk tolerance is much lower. This balance guides clinical decisions. Informed consent challenges. CRISPR therapies are complex. Patients must understand known risks, unknown risks, long-term uncertainty. True informed consent is difficult but essential. Equity and access issues. CRISPR therapies are expensive. This raises concerns about who can afford treatment, which countries have access, whether benefits are distributed fairly. Risk of genetic inequality. If access is limited, genetic advantages may concentrate. Health disparities may increase. This could create a new form of inequality, genetic inequality. Therapy versus enhancement. CRISPR could be used for disease treatment, trait enhancement. Enhancement may include physical traits, cognitive abilities, athletic performance. Most societies reject enhancement use. Slippery ethical boundaries. The line between therapy and enhancement is not always clear. Questions arise. Is increasing disease resistance enhancement? Is improving cognition treatment or enhancement? Clear boundaries are difficult to define. Dual-use concerns. CRISPR is powerful and accessible. It could be misused for unethical experiments, biological harm, unregulated human modification. This creates security concerns. Need for global governance. CRISPR science crosses borders. Ethical oversight must be international, transparent, enforceable. No single country can regulate it alone. Role of public trust. Public trust determines acceptance. Trust depends on transparency, ethical behavior, responsible communication. Misuse could damage trust for decades. Ethical summary. CRISPR forces society to ask, just because we can edit genes, should we? And if so, under what limits? Part 5. CRISPR beyond gene editing. Diagnostics, RNA, targeting, and gene regulation. CRISPR is more than molecular scissors. Most people think CRISPR cuts DNA. In reality, CRISPR is a programmable targeting system. It can recognize DNA, recognize RNA, bind without cutting. This enables many non-editing uses. CRISPR-based diagnostics. CRISPR can detect specific genetic sequences. If a target sequence is present, CRISPR binds. A signal is produced. No gene editing is required. How CRISPR diagnostics work. The system includes a guide RNA, a CRISPR-associated protein, a reporter signal. Binding triggers a visible readout. Speed and sensitivity. CRISPR diagnostics can detect disease in minutes, identify very low viral loads, match laboratory-level sensitivity. This makes them powerful point-of-care tools. Applications in infectious disease. CRISPR diagnostics can detect viral infections, bacterial pathogens, emerging diseases. They are especially useful during outbreaks. Cancer detection. Cancer often involves specific mutations. CRISPR can detect cancer-related DNA, identify minimal residual disease, monitor treatment response. This enables earlier diagnosis. Personalized diagnostics. Each patient's cancer is genetically unique. CRISPR allows mutation-specific detection, personalized disease monitoring. Diagnostics become tailored to the individual. RNA targeting. Instead of DNA, some CRISPR systems target RNA, not DNA. RNA editing is temporary, reversible, less risky. This offers safer therapeutic options. Advantages of RNA editing. RNA targeting allows short-term gene control, adjustable dosing, reduced long-term risk. Once treatment stops, effects fade. Applications of RNA editing. Potential uses include viral infections, neurological disorders, inflammatory diseases. RNA-based control is flexible and safer. Epigenetic editing explained. Epigenetics controls whether genes are active or silent. CRISPR can turn genes on, turn genes off, adjust gene activity without changing DNA sequence. Why epigenetic editing matters. Many diseases are not caused by mutations. They are caused by incorrect gene regulation, environmental effects, aging-related changes. Epigenetic editing addresses this. Reversibility and safety. Epigenetic changes are often reversible, less disruptive, ethically less controversial. This lowers clinical risk. Treating complex diseases. Complex diseases include diabetes, heart disease, psychiatric disorders. These involve many genes, not one mutation. CRISPR-based regulation may help. Research applications. CRISPR regulation tools help scientists study gene networks, understand disease mechanisms, identify new drug targets. This accelerates discovery. Beyond therapy. CRISPR platforms can be used for disease surveillance, environmental monitoring, precision diagnostics. Its impact extends beyond medicine. A new definition of CRISPR. CRISPR is no longer just a gene editor. It is a diagnostic tool, a regulatory system, a precision biology platform. Final reflection. The future of CRISPR may not be defined by how many genes we edit, but by how wisely we choose when not to.